So the live stream has begun. We have 6 minutes before we go live. So for everyone listening to us on YouTube, please just wait 5 minutes. Our webinar will begin shortly. Uma, could you just give a thumbs up if my audio is clear? Thank you. We will begin the webinar in 2 minutes. Okay, I am letting people into the webinar now. The webinar is now live. We have around 50 attendees, so I will begin the housekeeping instructions. Um, Uma, could you just please give a thumbs up if you are able to see the PowerPoint screen? Thank you, Uma. Um, so welcome everyone to our webinar. These are just a few instructions for all the attendees who are joining so that their Zoom call can go properly. The first thing that I'd like to talk about is the audio settings. So you can click on the audio settings in the bottom left hand corner here and make sure, and once you click on them, you will see this window pop up. So if we are able to invite you into the webinar so to ask a question on air, the audio should be working fine. The next thing is the raise hand icon. If there is time and time for it, you can click on the raise hand icon. It will turn green and your name will go to the top of the list, and the moderators will be able to speak with you, and they will be able to unmute you to ask the question. The next thing, which is most important for all attendees, is the Q&A box. The Q&A box is right here at the bottom, at the middle part of the bottom of the Zoom window. You can click on it, and this box will pop up, and you can type your question in. And once you've typed your question in, you can click send. You can also tick the send anonymously box if you do not want anyone to know that you have asked the question. And that's about it. And as far as Q&A goes, for those who are listening to us from YouTube, we will be monitoring YouTube actively, so please make sure that you type your questions there, and we will be relaying those questions into the Zoom call, where our moderators will select them. Then at the end, I would just like to say that there are no certificates whatsoever given for this program. So, and please do not mail us asking for certificates. Thank you. With that, I will hand it over to Professor Sudhir Krishna, who will formally introduce the topic of the webinar and begin the session. Over to you, Sudhir.
Thank you, Chandrakant. I'd like to welcome you all to the third public health webinar. I will not introduce the speakers and the panelists at length, as we have extensively circulated their profiles, and the registration numbers of 350 to 400 are a testimony to that. Before starting, just a sentence to say that our last webinar in the series, which will be a sort of mini online symposium about 6 weeks from now, where we will basically deal with the nitty-gritty of all these projects that you will hear today. So without any further ado, over to you, Dr. Jacob John. We're really looking forward to it.
Okay, thank you, Chandrakant, John Joseph, Shweta, Uma, and all the panelists and everyone. Greetings and a word of appreciation for organizing this webinar and inviting me onto it. I must warn you that there is a generational gap between me and all of you. You all are youthful, but I retired from my academic career in 2000, 20 years ago. So let me reminisce about my career in clinical virology. Next slide, please. Shweta, next slide. Okay, you know, when I was in medical school, in a good medical school, that was, bacterial cultures were very, very seldom done. All diagnosis was kind of clinical. The professor knew everything. Next slide. And in 1959, I came to Krishna Medical College for Diploma in Child Health, Pediatrics specialization. And I found here that bacteriology and mycology lab was open 24 hours. Cultures and antimicrobial sensitivity was routine. But there was no virology diagnostics. There was a separate research lab for arboviruses, which had detected for the first time in India Japanese encephalitis virus, dengue virus type 1, 2, 3, all kinds of goodies. Next slide. So post diploma in child health, I took 4 years of study leave from Vellore, spent 2 years in UK, finished my membership of the Royal College, MRCP Pediatrics, and then went to Denver, Colorado for a 2-year postdoctoral fellowship in pediatric infectious diseases. You must remember those days, infectious diseases was not a specialty either in UK, in a European country, or in India. Anyway, while in Denver, I dabbled a little bit in virology. I had 6 papers published in virology, 3 on vaccinia, and 3 on parainfluenza virus. And I managed the parainfluenza vaccine trial there. With my background like this, the medical school gave me gratis enrollment for master's in microbiology, and my work on vaccinia was accepted as the required thesis for the master's program. Next slide. So I returned to CMC Vellore in 1966, and I opened a diagnostic virology lab in 1967, the first in the country. We had many surprises, as you could imagine. When you look for new things, you find new things. But one of the most interesting was that we detected the oral poliovirus vaccine was not highly efficacious. Children could still develop polio in spite of 3 doses. Next slide. In the West, trivalent OPV efficacy was 100%. No child developed polio after even 2 doses. But we documented vaccine failure, and you can see on the graph, 3-dose vaccine failure range from 5% to 15%. But if you add 2 doses also range from 8 to 10% to 15 to 20%. That is unacceptable that you vaccinate children with the recommended doses and then still kids are not protected. Next slide. So I had liberal funding from WHO. In-depth studies were done on oral polio vaccine. 3-dose vaccine failure was rampant, and we designed many ways to improve vaccine efficacy. I will not go into details of many of those, except I'll show you one. Demand for virus diagnosis was high. Herpes virus in venereology, that is sexually transmitted diseases. Many problems in central nervous system diseases, and we found encephalitis caused by mumps virus, etc. Measles in sick children, and so much so we introduced measles vaccine in India. At a time when measles vaccine was not even licensed in India, we had to have special permission to import measles vaccine to children. Subacute sclerosing panencephalitis caused by measles, dengue and fever, rabies problem, lots and lots of problems were being solved. One breakthrough was, children get bronchopneumonia, children meaning infants below 1 year. And they all go on different antibiotics. But September through December every year, these bronchopneumonia cases were actually not bacterial, but bronchiolitis due to respiratory syncytial virus. When this was discovered, practice changed both in CMC Vellore and in the pediatric fraternity all over India, because they realized that bronchiolitis was common in India and often mistaken for bronchopneumonia. Next slide. Next slide. Okay. This is a summary of antibody response to trivalent oral polio vaccine. Type 1, the left cluster, type 2 in the middle, type 3 in the right cluster. Now watch. After 1 dose, 2 doses, 3 doses is the 3 bars, summary of many studies. About a third of children were only getting seroconverted with 1 dose. About 50% with 2 doses. About 2/3 with 3 doses were type 1 and 3. But type 2 was way ahead of type 1 and 3. If you really examine this graph, you will find something very interesting. This is not prime boost kind of response. This is arithmetic proportionality increments. The 29 to 50% rise, 50 to 65% rise is also 29% or 30%. 31 to 52, 52 to 67, again 30% increase. 67 to 89, 89 to 96, same proportion. So you could now predict as to what to expect with 4 doses, 5 doses, 10 doses, 13 doses, 15 doses. It would take about 15 doses to get 100% of children protected against types 1 and 3, but it takes only about 4 or 5 doses for type 2. Anyway, this had tremendous importance during the polio eradication days. Next slide. I'll show you one slide that even so, when we created an epidemic of polio vaccine in the community, which we called pulse immunization, you can see that 3 arrows going up in the bottom. That is 3 community-wide, irrespective of age, irrespective of past history of polio vaccination, all children below 5 getting 3 doses of vaccine a month apart. You look at, we had data for 2 consecutive years. So it's convincing. Look at 1982. The same thing in 1983, following 1982 last quarter pulse immunization. So here was proof that if you alter the epidemiology of polio using a vaccine and not building child by child by child herd immunity, you can have better results. Anyway, this is the origin of the word pulse immunization and the concept of pulse immunization. Okay, next slide. Okay, I'm shifting gears. The availability of a clinical virology department in a medical school enabled us to explore hepatitis B surface antigen in blood donors. Remember, those days it was called Australia antigen, discovered by Bloomberg in Australia, something weird. But the moment everyone realized that this was the cause of hepatitis B as post-transfusion hepatitis, all my colleagues in surgery and medicine said, we never see post-transfusion hepatitis. You know why? Because transfusion happens one day and it is 2 months, 3 months, 4 months, 5 months, 6 months later, somebody gets hepatitis. And who associates transfusion with the hepatitis? So we documented post-transfusion hepatitis. 1972 established routine screening for hepatitis B surface antigen. and showed that we prevented post-transmission hepatitis way back in 1972. It was popularized through Bulletin WHO. It became universally accepted. India made this legally required in 1989 only. Okay, next slide. All right, 1984, December 1985 early, HIV was discovered. Those days it was called HTLV3. AIDS became known in 1981, and lab reagents were created in NIH in USA to make sure that donor bloods are safe. Remember Arthur Ashe or Ashe, the tennis ace who got transfusion, HIV infection through transfusion. Anyway, we established a retrovirus lab in 1985. 85 detected HIV infection first in women in commercial sex, then in men with sexually transmitted diseases, then in blood donors, and even in antenatal women. Culturally, 99% of women here are monogamously married. 50% of men with sexually transmitted diseases in our venereology clinic are unmarried. Okay, so that is the cultural sexual behavior, just a window into the way commercial sex workers are utilized both by married men and by unmarried men, and they bring infection home. That's how women get infected. That is how antenatal women were exposed. Next slide. Okay, let me now shift gear totally and say, I'll describe 3 problem-solving experiences. I stepped in because I'm a virologist. It takes a virologist to recognize misclassified diseases called as viral infections, but not truly virus infections. Non-virologists think they are virus diseases. It takes an expert to say, hey, this is not a virus disease due to various reasons. I will not go into any of that. So the first problem was what was called Saharanpur encephalitis. Next slide. Now, in 1999, I was president of Indian Academy of Pediatrics. I heard of this disease in Western Uttar Pradesh called Saharanpur encephalitis. More than 2 decades, during September, November, December, young children died in large numbers in Saharanpur and in 9 or 10 surrounding districts with large outbreaks of encephalitis. Always young children of low-income families. Always well one day, but critically ill the next morning. Fever, convulsions, involuntary movements, slipping into coma. 70–80% case fatality. In hospitals that we checked, 800 to 1,000 children died every year. Pediatricians would tell me they shudder when the calendar changes from August to September because they expect large numbers of children with encephalitis. Okay, so I put together a team. Myself, the leader, a good pathologist to check the pathology, a good pediatrician to describe cases very carefully so that we would look at cases only with strict case definition, an epidemiologist, and eventually a toxicologist. This plant is an agricultural weed. It's called Cassia occidentalis. It kills animals if you feed Cassia occidentalis in their fodder, horses, cattle. Children were eating the green beans that you can see, the pods, and they were getting ill. Next slide. So we call that disease hepatomyoencephalopathy. Severe liver damage, temporary, recovers very quickly. Muscle necrosis, temporary, recovers quickly. Encephalopathy, the child doesn't die, the child recovers completely. So once this was unraveled, toxicology proven by a toxicologist in Lucknow, National Institute of Research on Toxicology, it was simple health education that stopped children from getting this disease or dying. Look at the numbers coming down from 2007 onwards in one hospital. And 2010 and 11, 00, 2012. Next slide. The team of the pathologist on the left, pediatrician in the middle, myself the third, and the epidemiologist on the fourth. We met for the second time physically. First time to plan, second time to celebrate. Next slide. Problem 2. Malkangiri district in Odisha. Southernmost district, mostly tribal. Next slide. 2016 November, I had an SOS call from the Odisha State Health Secretary saying that in Malkangiri district, there is an annual seasonal outbreak of encephalitis, causing death in many children. In 2016, up to November last week, 102 children had died. The Regional Medical Research Center experts were diagnosing this as Japanese encephalitis with very, very meager and questionable lab results. But the malariologist and the entomologist disputed that, saying that the JE mosquito is not here. All right. So we quickly, within one day, sorted it out. It was hepatomyoencephalopathy. Clinical features fitted, blood biochemistry fitted, CSF findings fitted, toxicology was able to show the Cassia occidentalis toxins in the urines of children with hepatomyoencephalopathy. With public education, no deaths in 2017, 2018. Next slide. Problem 3, Riddle wrapped in enigma in Muzaffarpur district, Bihar. Again, annually recurrent acute brain disease, known from 1970s, but first investigated in 1995 by the National Investigation Agency called National Center for Disease Control. Misclassified as viral encephalitis. Repeatedly investigated for all known viruses that could cause encephalitis, and then they were looking for unknown viruses for which CDC Atlanta people were called in. That is in 2012. 2013 is when I was invited, and it was solved in a very short time because as a virologist, I said, this cannot be a virus infection for various reasons. I will not go into that. Okay, in 2013, he said, hey, this is not a viral disease. Then what else is it? The biochemical disease. What biochemical disease? Next slide. Acute encephalitis syndrome was the name. It was actually an encephalopathy due to toxic origin. Published in Current Science in 2014. It had to be published very quickly because it would save children's lives. It had 3 etiological factors: undernutrition, prolonged fasting, sociological situation locally, and free availability of lychee because it's a lychee orchard area. And we worked it out. Then we had to prove 3 things. Is it actually encephalopathy? And the lychee encephalopathy is like Aki Jamaican vomiting disease, hypoglycemic encephalopathy. It's also similar to inborn errors of metabolism, acetyl-CoA dehydrogenase deficiency, that causes hypoglycemic encephalopathy following prolonged fasting. Okay, is it truly hypoglycemic encephalopathy? Because they had not been looking for such biochemical evidences till that year. Is there MCPG, methyl cyclopropyl glycine, which is an analog of methylene cyclopropyl alanine in active fruit, Jamaican? Is there amino acidemia, amino aciduria? All three were proved, the first two by us and the third by the CDC Atlanta people, because once they realized it is a biochemical disease, then immediately stepped in and completed the investigation. Next slide. Cases were clearly hypoglycemic encephalopathy. We did it in 2014, very early. The first 26 consecutive cases prospectively investigated all had blood glucose less than 40 milligram per 100 ml. When 10% dextrose was infused within 4 hours of onset, rapid and full recovery. Why 10%? Because unless you create hyperglycemia, the biochemical cycle that puts the child at risk of brain disease does not turn off. This is not merely hypoglycemia. This is hypoglycemic encephalopathy. Amino acidemia, these amino acids that are blocked elements of the incomplete cycle for fatty acid oxidation, complex issue. For virologists, it's Greek and Latin. Anyway, if you give 10% dextrose, children survive before brain damage takes place, which takes about 4 hours from start. Okay, something that everyone else had missed, which gave me the clue this is not viral disease. One of the clues. All children fell sick with what was called acute encephalitis syndrome between 5 a.m. and 7 a.m. Consistently for 10 years, season after season, everybody missed that big elephant. Next slide. So does lychee have MCPG? Toxicology friends proved that it had. Next slide. Lychee fruit contains methylene cyclopropyl glycine. Anyway, next slide. The proof of the pudding was no more deaths. Once this was unraveled, families were taught to feed the children before they go to bed. Lychee was restricted and 10% dextrose was given. And for the next 3 consecutive years, number of deaths were in 1-digit numbers, whereas it used to be 100 to 400 per year. Anyway, back to this slide. So the Department of Clinical Virology that I established also trained a number of people. And we decided that we will have a surplus of people and not, we don't have to sort of search for future faculty. So currently, the department has 5 professors, 1 department of clinical virology, not even microbiology. Microbiology also has about 5 professors. Why am I saying this? Because when ICMR could not find a director for National Institute of Virology in 2019, because the director retired, age bar, no suitable candidate for directorship. One of the 5 professors of clinical virology has been seconded from November 2019 for 3 years in NIV Pune. Just imagine if there was no director, and when does the coronavirus reach India? January 30. Just the nick of time. Dr. Priya Abraham, Professor of Virology in CMC, is the director in NIV. Now, when Indian origin scientists make it big in other countries, we all are elated, but we forget that they would not have been successful had they remained in India. Why? because our system is not made for achievement. We are not a result-oriented society. We are a process-oriented society. All the procedures are being done, ticked off one by one. But when it comes to system building, encouraging people to take risks in their career, be visionary, we are not up to the mark. Thank you very, very much. Sorry, I had to rush through because of time constraint, but I hope you have got the message all right.
Should I say? Thank you, Dr. Jacob John. That was absolutely fascinating. We will have you again as a panelist later in the session, and also, of course, 4 to 6 weeks from now. But we should move on. So, Dr. Jonna, would you go ahead, please?
Thank you for having me. I'm very pleased to be with you today, and I would like to now move us a little bit into what I think we can do for the future and how we can hopefully prevent ourselves from ever being in this tragic situation that we're in now. I'll talk a little bit about using the One Health approach, and Professor Jacob John was just very excellently, I think, illustrating some of the One Health approach when he was showing that he was bringing together the clinicians, the pediatricians, the epidemiologists, the virologists, the toxicologists, all coming together, also kind of using the social study in the home to be able to understand what was going on. And I think that is crucial to how we work for the future to make sure that we control epidemics at their source and prevent pandemics. I'm pleased that a definition of One Health is becoming more consistent and workable. And this definition is the one that I like to use, a collaborative, multisectoral, and transdisciplinary approach, working at all levels, local, regional, national, and global, with the goal of achieving optimal health outcomes. And really critically recognizing the interconnection between people, animals, plants, and their shared environment. And I'll just say that originally, as I've been working a couple of decades in this realm, the animal, human, and environment pieces were all there, but most recently, the plant health piece has been pulled out, used to be buried in the environment piece. And I think that's critical because plants are so instrumental to the health of our planet and our own health. Well, we are living in this tragic reality now with 22 million COVID cases and approaching 800,000 deaths worldwide. I show you here the kind of per capita map, but we are all living this tragedy and we're all in this together. And I think we can learn some things from some countries and regions that have fared better. And we can also, I guess, embrace what I think scientists have done so well during this COVID outbreak, and that is to work together, no matter what sort of political obstacles might be put in their way. The scientists are working hard to just span and recognize that we're all citizens of this planet and we need to work together. When we think about emerging health threats, and I'm going to speak primarily about viruses, we need to recognize that we're vulnerable, but we can do things to improve our situation. We are experiencing now on the order of 3 emerging infectious diseases of people every year, and that's actually increasing fairly dramatically. And the majority of those are spillover events coming from animal populations into humans. And as we continue to impact the Earth the way that we are, I think we're going to see that on the rise. That's what our modeling is showing us. All of the risk factors that point to the emergence events are increasing as our human populations are growing and changing the way that we use the planet, and especially pushing out to cover more of the planet with human habitation. So we are going to have these spillover events where a viral infection that is normal for its evolutionary host has the opportunity to actually spill over into other animals and make them sick, or into us and make us sick. But we can identify and stop these viral infections at their sources if we're ready, if we build the capacity, we open our minds and our awareness to not be so pathogen specific and not be so siloed, so that we're working together and we're ready. What we don't know is still the big number. So as I mentioned, and I'll describe to you a bit of a project, a proof of concept that gives me the hope and gives us the evidence that we can work differently and we can move into a new, safer realm. But just until recently, there were only about 240 known zoonotic viruses that could make us sick. But our data collection over the last decade shows us that actually there are about 500,000 viruses still to be discovered that can infect humans by spilling over from animal populations. Now, not all of those will make us sick. Not all of those are have pandemic potential, but we have the ability now. All of the tools are at our fingertips to be able to discover those, understand their transmission risks, mitigate those risks in communities with community support and education and participation. And we can start to build pipelines for better and faster diagnostics, vaccines, if we understand viruses better and understand why some really have the ability to infect and then go human to human spread. We can't understand that if we don't find them and we don't understand them and we don't study them. And that is, again, something that is completely feasible now, where it wasn't necessarily as easy to do 20 years ago, for example. So I'll explain to you now why I feel I can say that with some confidence. I'm not just prognosticating and waving my hands. I have been leading a project called the PREDICT project that's been funded by the US Agency for International Development. And it's been operating in 35 countries over the last decade, more than the last decade. So in 2009, USAID put out a challenge to the scientists in the US to come forward and really put our careers almost a little bit on the line to tackle a crazy challenge. And that was to start to preempt or combat viral infections at their source, at the first stages of emergence or spillover, and really to try to decrease the threat to public and animal health. Very difficult to design a project when you don't know what you're looking for, but we, some of us came together and responded to that call because we felt that it was, the time was appropriate and we had the tools and we could think about viral diseases in a different way rather than siloed pathogen by pathogen. Because doing that pathogen by pathogen means that we wait until the virus spills over, gets into the human population, and frankly is killing enough people that we even notice it. And that's basically the terrible situation that got us to this place with the COVID pandemic. So what do we believe we need to do? We need to understand the viruses as they are in this zoonotic pool in their evolutionary host, and be ready for them before they spill over preferably, but if not, we could pick them up as they're amplifying in domestic animal hosts and people early and control them more quickly at their source. So to do that, we need forecasting and readiness. We need early detection or laboratory capacity. And hopefully that allows this rapid response that lowers those curves, because prior to this, we spend most of our outbreak response efforts really after it's too late. And I think we can all understand this much better now, given this situation we're living in. Most of the world still in and out of sheltering in place and quarantining, and the secondary wave of the pandemic, which is the mental health, anxiety, isolation that comes from these horrible consequences of having to live without a vaccine, without, unfortunately, the good public health practices early on because of lack of diagnostics right at the beginning that could have allowed better control early. So the PREDICT project was formed by responding to this call from USAID and a group of organizations in the US that I've shown here got together to put in a bid to USAID. But most importantly, after we were the successful bidders, what we did was we went and used this One Health approach to bring together the Ministries of Health, Agriculture, and Environment in the countries where we were able to engage. Now, we only engaged in 35 countries. We had a very kind of small, for the problem size, a kind of small portfolio of where we could engage, but it did allow us to provide a proof of concept that this work could be done, this virologic work and epidemiologic work could be done on a larger scale. So we primarily focused for much of the time on the highest risk taxa for viruses to spill over to people, and those were non-human primates, bats, and rodents. But we also were able to work with FAO and Ministries of Agriculture to look in livestock, and with WHO and Ministries of Health to look in humans, especially those humans that were having intimate contact and increased risk because of their occupational exposures or the way that they're culturally were interacting with animals. And then we were also able, kind of as Professor Jacob John mentioned, to work on these investigations where we worked in the clinics with people of fevers of unknown origin and looked to see what viruses they were sharing with animals in their communities. In fact, the PREDICT project worked on more than 50, almost, I think we're up to 60 outbreak investigations in the countries where we worked over the decade. And the work was not so glamorous. It was a hard roll up your sleeves work. I think you heard it referred to as the nitty gritty. I guess I would say we swabbed a lot of orifices, a lot of butts, if you will, to find the viruses. So we were only looking at these high-risk interfaces. I show you some here, including, as you see, the camels and the chickens, or the chickens and the pigs, where we're thinking about influenza or MERS. We are looking for the known pathogens, but we're also looking at, for the unknown pathogens, the discoverable pathogens or potential pathogens. And they're also in the species that you can't see there, including the rodents and the bats that are in these same interfaces and mixing. In the middle there, I have a bat guano attractant that is a large industry now, developing industry in Southeast Asia, where people are attracting bats to their farms, is collecting the guano, putting it directly onto their farms, or taking it to market and selling it, putting themselves at risk for what the bats might be excreting, but potentially there are some good practices that could allow some of that to be safe, but none of that had been investigated. So I'll just quickly go through. I know we want to have time for discussion, so I'll quickly go through and just mention that during the project, we have been able to discover about 1,000 viruses, and at the same time, using a kind of more agnostic or broad-based approach where we were looking for viral families rather than for the actual viruses that are known to be pathogens. We could identify those new pathogens or ones that we don't yet know if they're pathogenic, but are in viral families that have been known to cause epidemics and pandemics. But with the same platform, we were also at the same time identifying known pathogens, allowing countries, communities, regions to realize that they had pathogens that they didn't know that they had already in their region, and identifying the new hosts and the interfaces so that they could start to get a handle on their risk and mitigate and reduce behaviors for that risk. So that's an interesting finding, but I think the more important findings are kind of on the top row of this graphic, and that is that we indoctrinated, if you will, almost 7,000 people, trained people in ministries, in academia, in research institutes, and even policy folks to really be ready for these outbreaks and to use the One Health approach. By doing that, we worked in more than 60 laboratories in the countries where we engaged, so that those laboratories had the ability to do viral discovery, again, without really incredibly expensive equipment like next-gen sequencing equipment, which we also use, but we used on a smaller scale to be able to compare sort of traditional consensus-based PCR at the viral family level, then with full sequencing to next-gen sequencing to see how we could optimize viral detection and discovery in some of the most resource-limited locations. And what did that do? Did we find COVID? We did not. We were not working in the area of China where SARS coronavirus 2 appears to have emerged. We were working with Chinese colleagues, but primarily at the Southeast Asian border in southern China in the Yunnan province. But we discovered a huge amount of information, including more than 100 coronaviruses, that led us to be able to aid in beginning to understand what hosts might be contributing to SARS coronavirus 2 ecology, as well as epidemiologically risks that we identified in China, in Southeast Asia, in other countries where we work that put people at higher risk for exposure. And in fact, we were able to document exposure to SARS-related coronavirus exposure throughout southern China. And it was related to behaviors like working in live animal markets or working with animals, including wildlife farming. In fact, the highest risk chain that we identified in Vietnam was that wildlife farming and butchering all the way up to live wildlife restaurants presented the highest risk in Vietnam for exposure to coronaviruses. So while we weren't able to identify SARS coronavirus 2 ahead of time, that's going to take a bigger project, and the one that I'd like to talk to you about today, and hopefully still your interest and also solicit your support and action. But we were able to work and rapidly reactivate the folks in the countries where we had engaged during the PREDICT project. The PREDICT project had been slated to end last September, but because of the terrible outbreak, we were able to be reactivated. And we saw our teams, even before they were reactivated, jumping into action in places like Nepal and Thailand, diagnosing the first cases of SARS coronavirus 2 or COVID-19 disease coming into their countries using this consensus-based PCR platform before there was a full sequence known out of China, which our China PREDICT country coordinator was the one to release that that sequence so that better diagnostics, more specific diagnostics for SARS coronavirus 2 could be developed. But even before that was known, the other country teams jumped into action. They identified cases in their countries so that public health procedures like contact tracing and quarantining could be done and put those countries in better conditions than many other countries. In Africa, our teams also became technical assistants for their countries. Some of our folks were the first people in their countries to publish on coronaviruses because of the PREDICT project, so they were the experts in their countries. And in all of the countries, they are helping to do testing now, ruling in and ruling out cases. So we're very proud of this proof of concept that when the laboratories, those 60 laboratories that I mentioned, are ready, the countries are better prepared and can jump into action very quickly. So we didn't find this one, but if we had taken it to scale, and this is what I think we can do now, if we can go to identify all those 500,000 viruses that are out there and available to spill over into people, how much better off would we be? Well, some of the information that you get out of collecting these large catalogs of viruses, and I'll use still the SARS-CoV-2 example, is that, for example, with our discovery of these SARS-related coronaviruses, I mentioned we've discovered 100 new ones, but we also found them thousands of times in many species. And we could start to identify the most closely related ones were in which species. So we could say the SARS coronaviruses here were most often found in rhinophilid and hippocytarid bats. That allowed the folks working in China to be able to start to target what's the likely evolutionary host that allowed the first spillover. It might have spilled over into another intermediate host, kind of like the first SARS, we believe, spilled over from a bat to a civet and then amplified. But it started to give the clues. And this is the kind of information that we should know ahead of time. We should know what the risky species are. We should know where the viruses are. If we have that catalog with the risk information ahead of time, we can be prepared and we can know exactly where to look and to start to get things under control even more quickly. I do want to say that we need to protect bats because I often am talking about viruses that spill over from bats. And I want everyone to remember that bats are incredibly important for biodiversity and our ecosystems. They control pests, they pollinate, they connect fragmented landscapes by dispersing seeds. So as we continue to convert our landscapes where there's just pockets of endangered plants, we need to connect them. But also if you kill bats, it's generally an unsuccessful way to control their populations because they increase their recruitment. And what we know, because again, the PREDICT project has documented when bats are shedding coronaviruses, that actually it's that recruitment and increase of birth rate that increases the number of susceptibles as well as the shedders, because they tend to shed coronaviruses as they're being weaned from their mothers. So what can we do with this information? When I say take it to scale, what could we do? Well, we could, I'm telling you, if the world joins together, we could, in 10 years or less, we could identify almost all of the viruses, if not all of the viruses available to spill over to us, and understand where they are, in which hosts, and what their potential for epidemics and pandemics are. Again, all of that information is at our fingertips, but what do we need to do? We need to mobilize a large workforce to test every mammal and bird, or at least water bird species in the world. We need to do that at large numbers. Our data show that we need to test about 2,000 of each species in each ecoregion to be able to find the viruses that are circulating. And as we find more viruses, we start to saturate the viral discovery curve, and we can tell when we're getting close to finding all the viruses in those species. And we've mapped that out because of the PREDICT project. The PREDICT project allowed us to also invest and develop all of the more than 20 protocols and guides to do this safely for us, the samplers, but also safe for the wildlife that we would sample. Then we need to get into those laboratories, as I mentioned, but the technology is very accessible to identify those viruses. And then we need to inform strategies and prepare and prevent spillover. Now, that is something that is going to take political will as well. So we need the science to be well respected and to be valued, and then engender the political will to take action to be prepared. And if we don't have the justification now based on this COVID-19, I don't hold a lot of hope for humankind. We must learn from these experiences that we can't wait for the next outbreak, because this one could have been so much worse than it was as far as its deadliness. So we can pull together this global network and share data. So this doesn't have to be one huge project. Every country, region, NGO that wants to contribute could contribute, as long as we're using some of the same protocols and collecting at least the same minimum data and sharing it. That will allow us to actually evaluate the risk. And again, because of the PREDICT project, we've been able to start that process to develop a risk ranking for the viruses that we found in PREDICT. It allows us to pull together not just the virologic information, which is sort of the traditional way to rank risk, but to also pull together the host and environment data. And what we're finding is those viruses that have the ability to infect multiple hosts, and so we find them in multiple hosts through a project like this, those are the ones that tend to have the highest potential for infecting humans and going human to human spread. So what will it cost? It's a big price tag. I'm not going to try and minimize that. We need, if we were to really try to discover all of them, we need about $4 billion US to do this all over the world, but distributed across 10 years and across all the countries and investors that would be willing to contribute. Now, that is a big sounding number, but when we compare that to the $4 trillion US that this pandemic is costing, it's minuscule. If we compare it to even one of the large outbreaks and epidemics, large-scale epidemics that came before it, like the first SARS, we're still talking about less than 10% of a single outbreak to discover and understand all of the risk that is out there at this time. So we really believe that as we pull together, and we can get into, as has already been mentioned, in the 4 to 6 weeks from now, we can get into the nitty gritty of how to do this work. But if we pull together, we work together, we can start to understand so much better and control our own fate by really getting that risk paradigm, improving surveillance, working with vulnerable communities where we know that the behavior practices are putting them most at risk. We also will start to fuel that pipeline for drug development and rapid vaccine development pipelines. And we really learn a lot about our planet, which is so important to conserve, because as we've been even doing the PREDICT project, we've been able to identify new cryptic species that weren't necessarily known to mankind beforehand, because we're doing not only the virology, but the genetics of the hosts as well. So with that, I will hand it back.
Thank you, Dr. Jonna. That was very informative, and we'll be back again on the panel discussion. We'll just move on right now with Dr. Thumbi Ndung'u. Dr. Thumbi.
Good morning, everyone, and I'd like to to thank the organizers of this meeting for inviting me to present. I'm going to be talking about HIV, which, as some of you know, is actually a zoonotic disease. And so, although I'm presenting in this One Health forum, I do actually feel at home because of the fact that the HIV originates from animals, and also I'm a veterinarian myself by profession, and so I'm going to be approaching this, with that reassuring background in my mind, even though I'm going to just be talking about HIV in humans primarily. So we all know that HIV infections have been going down globally steadily over the last decade. And this is primarily as a result of antiretroviral therapy. The fact that there has been extensive rollout of antiretroviral therapy throughout the world. But however, we are going to be missing the 2020 targets for new HIV infections that were set by WHO and UNAIDS. We also know that HIV infection is associated with a lot of comorbidities in people that, even in people who are on antiretroviral therapy. So individuals who have HIV infection, even when they have very well controlled infection, have at higher risk of developing cardiovascular disease, metabolic disorders, neurocognitive abnormalities, malignancies, and they have also reduced life expectancy. So overall, HIV incidence remains high worldwide, particularly in some population groups, and current prevention strategies are suboptimal. So we need to do much more to develop new technologies and new approaches to controlling HIV infection. And even if we were able to reduce new HIV infections, we still have significant HIV-associated multimorbidity in people who are already HIV infected. And we need to find a way to deal with this long-term consequences of HIV infection, as well as long-term antiretroviral therapy usage. So I wanted to present some data from South Africa. I come from South Africa, which, as most of you may know, is at the epicenter of the HIV/AIDS epidemic. We have one of the worst HIV epidemics in the world. And this data here just shows high incidence of HIV infection, particularly in young people. You can see that among young people between the ages of 20 to 24, HIV incidence is approaches about 8% per 1,000 person years, which is extremely high. And furthermore, there is this data from a surveillance project that we have been undertaking in rural KwaZulu-Natal. And you can see that in this particular population, you have very high prevalence of HIV infection, particularly in individuals between the ages of 25 and 44, where the percentage of HIV infection is about 36% among males and close to 60% in females, extremely high rates of HIV infection. And I wanted to use this data to illustrate 2 points. One is that we are failing in stopping new HIV infections. We still have extremely high HIV transmission in some communities, certainly not in all. But the other point that I wanted to make is that even if we were able to, again, solve the problem of HIV transmission, you still have certain populations that have very high rates, high prevalence of HIV infection. And we are going to have to figure out a way to deal with this high burden of HIV infection in some communities that is long life, considering the fact that at the moment, we cannot cure HIV. Now, this is despite the fact that we have very good tools for HIV prevention that have been shown to be effective in clinical trials, in randomized clinical trials. So we know, for example, that control of sexually transmitted infections, HIV education and counseling, empowerment of women, the use of condoms, male circumcision. All of these are, proven tools for prevention of HIV infection. And I should also add, of course, pre-exposure prophylaxis. But these tools are suboptimal, and we need to be thinking about developing the next generation of tools, particularly ones that have been shown traditionally and historically to be very effective in control of infectious diseases. And some of these, for example, include vaccines. Obviously, we don't have an effective HIV vaccine, but we know from studies of other diseases that a vaccine would have the most significant impact in reducing HIV transmission. And then there are other approaches such as passive immunization, where we can administer, for example, monoclonal antibodies that can prevent infection. And we also need to think about other novel approaches that we can develop to add to the current toolbox in terms of HIV prevention. So in our setting here in South Africa, in Durban, in KwaZulu-Natal, which is really the epicenter of the HIV/AIDS epidemic, because as I've shown you, we have very high rates of HIV infection. We have been undertaking a project that where we try to identify people with acute HIV infection. And the idea of this project is to see whether we can develop new and innovative ways of stopping new HIV infections. But in the instance where somebody gets HIV infected, we are also interested in understanding what is it that we can do very early in the course of HIV infection to really either cure HIV infection or eradicate HIV infection from the body. So we try to identify individuals at the earliest stages of HIV infection, what we call phoebic stage 1 and 2. This is before humoral immune responses develop, but when viral load is detectable. And so we do this by screening for HIV RNA. And we try to identify people before they reach peak viremia during acute HIV infection. And some of the key questions that we have been interested in answering are: what are the characteristics of the transmitted founder virus? And we're also interested in understanding immune responses in acute HIV infection, because we think that this could be a pathway to developing either effective vaccines or modifying the natural immune response so that it can durably control HIV infection. And then, of course, we have also been very much interested in the question of what is the impact of early HIV treatment? What happens if you are to treat somebody very, very early? How does that change the trajectory of disease? And can this actually lead to eradication of the virus from the body? So the ultimate goal of our work is to develop new or improved HIV prevention strategies, as well as to see whether we can actually achieve a durable control of HIV infection through cure strategies. So this study, we call it the FRESH study. And FRESH stands for Females Rising Through Education, Support, and Health. And what we do in this study is that we recruit young women between the ages of 18 to 23 who are at high risk of HIV infection. Like I said, this is a region where HIV infection rates are very high, but also these are sexually active women that we recruit into this cohort. We provide them with an intensive HIV prevention program, because the idea is to really prevent new infections. So we provide them with all the technologies for prevention of HIV infection, including pre-exposure prophylaxis, and also life skills and job readiness curriculum. And they come to our clinic twice a week. But when they come to the clinic, we do a finger prick blood draw. And we can use this finger prick blood draw to test for HIV RNA. which therefore means that because these women, when they come into our program, they're HIV negative. If somebody becomes HIV positive when they are on the program, we can immediately detect HIV infection through RNA testing, and then we can initiate antiretroviral therapy. So I'd like to present some of the data and some of the lessons that we have learned from this particular study. And first of all, I'm going to talk about the microbiome. We have been interested in the microbiome in HIV negative women in this cohort. And we published this study a couple of years ago showing that there are significant differences in the female genital tract microbiome. And you could actually classify the women into 4 cervical types based on the diversity of the microbiota in the female genital tract. And as you can see here, in cervical type 1, the women have a dominant Lactobacillus crispatis, whereas in cervical types 3 and 4, for example, they have a much more diverse microbiota. And so the microbiome actually differs significantly from one woman to another. And what we found is that in fact, having a Lactobacillus dominant microbiota was associated with low levels of genital inflammation. I'm not going to show you all the data, but I'll summarize it and say that having a Lactobacillus dominant microbiome was associated with low levels of genital inflammation. And in fact, these women were less likely to acquire HIV infection compared to women that had a much more diverse microbiome, for example, those in cervical types 3 and 4. And so based on this study, we are now beginning to investigate whether we can actually alter the vaginal microbiome of women by administering a probiotic formulation known as Lactin V. So the hypothesis that we're going to be testing in the upcoming study is whether the use of Lactin V by young South African women will decrease genital tract inflammation that is associated with increased HIV acquisition. Now, Lactin V is a probiotic formulation, as I said, that is dominated by lactobacilli that has already been shown to prevent recurrence of bacteriovaginosis in a paper that was published earlier this year in the New England Journal of Medicine. And the aim of this study will be to determine the effect of intravaginally applied Lactin V on genital tract inflammation, to determine the ability of Lactin V to promote a Lactobacillus dominant vaginal microbiota, and of course to determine the safety and acceptability of this product as a way to increase the tools that we have for preventing new HIV infections in this setting. Okay, I want to talk a little bit about what happens when some of the women in our cohort do get HIV infected. So one of the things that I said is that we provide antiretroviral therapy immediately once we detect acute HIV infection. So this is, some of these women have been started on antiretroviral therapy as early as 1 day. In fact, on average 1 day after detection of acute viremia. And you can see that the first 14 women here that are shown in the black line were not treated with antiretroviral therapy immediately, because at that time we were following the South African guidelines for treatment of HIV infection. And treatment was only initiated when CD4 cell counts fell below 350. But for the women shown here, represented in the blue and red lines here, we started antiretroviral therapy immediately. And in the case of the red line, we used the 3-drug regimen that is the standard of treatment in South Africa, plus we intensify the treatment with an additional drug, raltegravir, to see whether we can actually reduce the reservoir further by introducing 4 drugs. And you can see from this graph that by intensification with raltegravir, we actually reduce the time to full viral load suppression in these women, so such that within about 2 weeks of initiation of antiretroviral therapy, all the women become pretty much undetectable in terms of plasma viremia. So we were able to show from these studies that early treatment blunts peak viremia. And this is data shown here, and that early treatment also preserves CD4 T cell counts. So we think that this is good for preserving the immune system in HIV infection. And with other data that I'm not going to show, we were also able to show that early treatment actually prevent seroconversion. So the women do not actually even seroconvert because we are starting antiretroviral therapy very, very early. Now, we have been interested, as I said, in using this cohort to see what are the interventions that we can put in place for either prevention of HIV infection or for treatment and eradication of HIV through cure strategies. And one of the strategies where we are very interested in exploring is the possibility of using broadly neutralizing monoclonal antibodies, either through passive immunization to prevent new HIV infections. And so one of the things we have done is to sequence the transmitted founder viruses from this this cohort of women infected in this cohort. You can see from these phylogenetic trees that most of the women are in fact infected with very homogeneous virus clones. So suggesting that only one single virus infected individuals in this cohort. So and we know that this is not a new finding. It's actually quite well known that HIV infection is established by one single variant because there's a significant genetic bottleneck when HIV is transmitted, such that only one single variant of HIV establishes infection. We have then gone on to test whether these transmitted founder viruses can be inhibited by broadly neutralizing monoclonal antibodies that are available in the field and that are now beginning to enter clinical trials, either for prevention of HIV infection or for treatment. And it's quite interesting that these transmitted founder viruses, for example, in our cohort, you can see here that they are not very sensitive to this particular monoclonal antibody known as VRCO1. So for those who are not familiar with these heat maps, the more intense the color here, like the intense red color, shows that the viruses are very sensitive to the broadly neutralizing monoclonal antibody, and the lighter the color, the more resistant those viruses are. So you can see here, for example, that VRCO1 is not a very good monoclonal antibody, despite that this monoclonal antibody is now in clinical trials here in South Africa to see whether it can prevent new HIV infections. But then we have other monoclonal antibodies like the one shown here, VRCO7, which is very good at inhibiting most of the transmitted founder viruses in this cohort, as well as this particular monoclonal antibody known as CARP256. So essentially what we believe is that we think that using an approach such as ours, we can identify some of the next generation tools, such as monoclonal antibodies that are likely to work against current circulating viruses in our setting. And this would enable us to then use these monoclonal antibodies either for passive administration to prevent new HIV infection in the absence of a vaccine, or we may be able to use them for therapeutic purposes for curative, for cure strategies within our setting. So we are planning now to use this cohort for proof of concept human studies for functional cure. And our hypothesis is that because we are starting antiretroviral therapy very, very early during acute HIV infection and preserving the immune function through early intensified antiretroviral therapy that is started during the acute phase, that this could be a pathway to achieve HIV remission, particularly if it is combined with latency reversal agents, such as toll-like receptor agonists, as well as monoclonal antibodies, broadly neutralizing monoclonal antibodies that can penetrate into the reservoir sites where the virus hides. But these monoclonal antibodies also have potent antiviral action against these transmitted founder viruses. And so we think that this is a plausible approach to achieving an HIV remission. We don't know for sure that this is what is going to happen, but this is what we are planning to do as a proof of concept study for a cure strategy that might be relevant to an HIV endemic area such as ours. And there are other approaches out there that are now being explored that I just wanted to mention, such as vaccines, as well as use of immune modifying agents to see whether we can actually eradicate HIV completely, or at least achieve long-term remission that would be useful for HIV eradication in high burden settings. And then I wanted just to mention a little bit about some of the COVID-19 research work that we are doing within this region. And of course, we have very high HIV burden, and also TB is a problem in this setting. So some of the questions that we are interested in addressing are how is the course of COVID-19 disease different in HIV-infected individuals on antiretroviral therapy or off antiretroviral therapy compared to HIV-uninfected individuals, and how does this affect potential protective immune mechanisms? How do TB and other respiratory infections or lung damage from TB and these virus diseases affect COVID-19 disease? Why are children relatively protected from severe COVID-19 disease? And also we are interested in addressing whether HIV or TB affect vaccine or therapeutic effectiveness in COVID-19 disease. Finally, I wanted to just talk a little bit about some capacity building activities that we are involved in. And I'm aware that most of the people attending this webinar are young people. So I thought that I should start off with this slide here that shows the numbers, the percentage of people that below the age of 35 that went on to win Nobel Prizes in different fields of science, in physics, chemistry, and physiology and medicine. And what you see is that most of the people who went on to win a Nobel Prize actually won a Nobel Prize for work that was done when they were under the age of 35. And I can almost bet that most of the people attending this webinar are perhaps in this age group. And so I just wanted to say that you are in the right place at the right time if you are a young person at this time, because the opportunities for creativity are enormous. And the age of productivity is actually mostly below the age of 35. And so you have a tremendous opportunity to make a significant contribution to science at this particular time. And it may not be that you go on to win a Nobel Prize, but certainly you can make a significant scientific contribution, particularly if you are in the younger age groups. So with that in mind, I wanted to very briefly discuss about an initiative we have here in Africa. We call it the Sub-Saharan African Network for TB and HIV Research Excellence, SANTE. This is a project that is funded by the Wellcome Trust and the African Academy of Sciences. This was funded about 5 years ago, so the program is coming to an end. We have had about more than 100 trainees at master's, PhD, and postdoctoral level. Our overall goals in this program are to strengthen South-South partnerships in HIV and TB research, to train the next generation leaders of African science, and to create enabling environments for excellence in research and collaboration in Africa. Our specific aims, and I will highlight some of our achievements, was to establish a program of research on HIV and TB that is multidisciplinary towards prevention treatment of HIV, and we don't discriminate between the disciplines. So we are just interested in good science, no matter where it's coming, the discipline that is coming from. We have had more than 100 publications, including publications in science, Nature Communications, HIV, Lancet HIV, and some have gone on to really actually influence policy. We have offered a training and capacity building program across 12 African partner sites. We have had more than 100 trainees at various levels. Our goal was also to build a very strong institutional network for research excellence. I think we have achieved this by having attracted more than $15 million of additional funding that we have leveraged over the course of this grant. And of course, we have also had a very strong program in community and public engagement. And we have very strong public and community engagement across our network now. So just to conclude, I would like to say that optimization of current proven approaches for HIV prevention, as well as innovations to develop new approaches such as vaccines, is needed because the global burden of HIV remains extremely high. I think we need to be looking at new opportunities for HIV cure or achieving long-term remission of HIV, particularly immune-based approaches in resource-limited settings. And then, like I said, there is a tremendous opportunity, I think, for capacity building and South-South collaborations. And these are absolutely necessary for both for epidemic preparedness, but also to improve the health and economic development in resource-poor settings. And finally, I just wanted to acknowledge my colleagues. Obviously, I don't do this work alone. I have a team of very dedicated colleagues, and it is my pleasure to be working with all of them. And I wanted to thank them, as well as our funders who generously fund our work. Thank you again for listening to me and for giving me the opportunity to be part of this really exciting webinar.
Am I on? Yeah. Well, thank you. That was very inspirational. So Uma, will you take over a panel discussion? And if you and Shahid could try and keep it, as Jonna said, somewhat action focused, so that I mean, I think Jonna made it very clear that this is the time to act. So let's start acting, and will you start the process, and we'll follow it up. We'll also get all our funders on board, the government funders, Narayan Murthy, Nadir Godrej, all the people that we're talking to. So start the process.
Thanks, Sudhir, and thanks to all of you for your talks. Maybe I'll invite Shahid to start off with some comments, and there are a few questions. They aren't action-oriented specifically, but very interesting. So maybe I'll ask Shahid to comment first, and then I'll bring some questions in, if that works, from the audience.
Yeah, can you hear me, Uma?
Yes.
Okay, yes. Wonderful. Thanks to all the speakers who really gave wonderful talks and focused on the importance of what we're trying to do today. Let me go back to the Middle Ages and remind you that diseases that killed a whole lot of people were all bacterial diseases, and plague was really the biggest one. But since the development of antibiotics, you don't hear about bacterial diseases killing as much. If you look at the 20th century, and the big one that came in the 20th century was the Spanish flu. Of course, it didn't originate in Spain, so it's still an enigma why it's called Spanish flu, but nevertheless, for a long time, Spanish flu was believed to be plague because of the memory of the late 19th century plague that was still continuing. But there were two other pandemics of flu in the 20th century, the Asian flu of 57 and the Hong Kong flu of 68. And then we failed to recognize that there was another pandemic that was started in 20th century that still continues. And this is something that Thumbi just talked about, and that's HIV. HIV is a continuing pandemic. As we moved into the 21st century, in the first 2 decades of the 21st century, there have already been 2 pandemics, swine flu in 2009 and COVID, which is going on right now. But there have been many near misses, something that Jonna talked about. We have SARS, we have MERS, we have Zika, we have flu H7N9, bird flu, Ebola, Zika. And I'll also remind you what Jonna said, that there are about 3 new emerging infections that come to notice every year. Of course, all of them don't become outbreaks of concern or even epidemics, but this is something to worry about. So why I'm telling you all this is that the frequency of emergence of new infections, of new diseases has increased over time. The other notable thing to see is that in all the pandemics of the 20th and 21st century have been caused by viruses. Viruses comprise about 14 or 15% of known human pathogens, but they contribute to almost 50% of new emerging pathogens. And that's why meetings such as this, consortia such as this, make a whole lot of sense. If you looked at the mammalian species that harbor zoonotic viruses, and you do a plot of percent zoonotic viruses on the y-axis and percent mammalian species on the x-axis, you will find that rodents, followed by bats, followed by non-human primates. These are the ones, and of course, I talked about mammalian, but birds are also important. So these are the ones we need to focus on. But if you also look at the most abundant species on Earth, guess what? Humans. So essentially, we are the substrate for everything else that is incubating in all the animal species. So be aware of that. What do we have to be worried about? We have to be worried about our ecology, not destroying forests at the speed at which we are destroying them. Be aware of climate change, and there have been modeling studies done. In fact, studies done from the department that Jonna comes from, which have shown that if you stuck to the Paris Agreement, or if you didn't stick to the Paris Agreement and let the temperature on Earth go haywire, how new diseases will move into new locations is rather scary. So people in the Northern Hemisphere, as far north as US and Canada, the northern parts will start experiencing dengue much more than they experience today. And finally, I will remind you again of something that Jonna said, of the cost of all this. If the Global Virome project over 10 years costs $4 billion, please be reminded that SARS in 2009 SARS in 2003 cost $40 billion, 10 times more than this. And the estimates for COVID are somewhere around $4 to $5 trillion. Having said that, these are very tough times to raise funds, simply because the global economy is in a downturn. But on the other hand, these are very good times to raise funds, because everyone, a virus is a household name now. And frankly, I didn't realize that there were so many virologists in India until I realized that the next week they became legal experts and the following week, they became military experts. So we are living in a sensitive and sensitized world. And this is probably the best opportunity we will have to gain the ears of both the political leadership as well as philanthropists. So all the power to this group. I'm happy to be part of this. Thank you.
Thank you. So we heard a lot of very interesting talks, and we heard about, from Dr. John about the need for capacity building, and this program is critically involved in that. We also heard about that from Jonna in terms of the international efforts at capacity building that Predict has engaged in, for example, and also from Dr. Thumbi about how even ongoing epidemics require capacity of various kinds. So I'm just curious about asking all of you, whoever would like to, my one question, and then I'll bring up the audience questions. The task is definitely large, and it's not just about money. So how do we, and it's also challenging because it's a distributed task. It's very different from, scientists. We may be, if anything, able to grapple with tasks which are focused and in one place. But these types of distributed activities is something which is hard. And in some sense, given the habitats and diversity of available infrastructure and so on in India, India itself poses many challenges. So maybe Dr. John and Dr. Thumbi and Jonna, from your experience, a couple of lines or sentences, and also Shahid, on how you think India can specifically approach, should there be an interest in engaging in such an activity.
Hey, you want me to go first?
Yes, go ahead, please. Thank you.
At a time when the COVID pandemic should have brought the whole world together and working together because it is a global problem, same thing, every country. What did we see? We saw kind of a fragmented, unorganized kind of response to the pandemic. Even WHO leadership was not as visible now as in the time of SARS. WHO leadership was fantastic. So we have a human disease that is, we do know a lot, but that does not motivate us to behavior change. And knowledge does not protect. Behavior change does protect. How do we transition from, even money can be raised, how do we transition into a kind of a human response, cohesive response to problems that everyone has pointed out as the wolf is at the door actually, but we don't seem to take the wolf very seriously. Let me stop by saying that.
Thank you. Jonna?
Well, I agree with that, and there's been lots of disappointments in this one. Disappointments that warnings weren't heeded, but human nature is such that we always deal with what has just happened to us instead of what is coming next. And we clearly can't afford to do that any longer. I think India is such a, from my outsider perspective, but with colleagues and friends like you, but is such a innovation and technological and intellectual powerhouse for the world right now. It also, for doing this kind of work, has all of the hot spots kind of issues. And one of the questions was really around hotspots and the big things that show us in our modeling that are related to disease emergence, especially viral disease emergence, and our human population growth, biodiversity, so the number of habitats, the number of species, and then some of the other drivers, especially land use change and climate. So because all of those things come together in India, I can't imagine a more equipped and important place to do this kind of work, really.
Thank you. Dr. Thumbi?
So thank you very much, Uma. And so I have to start by admitting that I don't fully, obviously, appreciate the landscape of science in India and the way it is approached. But I'll talk in general terms about my perspective. I think that what the current epidemic, and even an epidemic like HIV, which continues obviously to persist, teaches us is a need for us to have a multidisciplinary, cross-disciplinary approach to science. And I think that for too long, in my opinion, science has been approached from a very sort of discipline-specific, very sort of compartmentalized silos, of microbiology, epidemiology, and so on. And so I think one of the challenges that we have, in fact, in my opinion, is that to change our approach to how we, as scientists, approach the problem of science and solving problems in society. That the kind of challenges that we currently face call for a multidisciplinary and cross-disciplinary approach if they are going to have societal impact. And even in the way we train students and the way we develop young scientists, emerging scientists, I think we need to change and recognize that and see how can we improve collaboration without compromising excellence, because we do want to continue to promote scientific excellence and innovation, but how can we do that without compromising excellence, so that we are promoting more collaborative scientific excellence rather than individual, sort of scientific, sort of bright shining stars that are kind of operating in isolation. So I think that's one of the challenges that we have and that we need to grapple with. And then the second one I wanted to quickly raise was, in my opinion, is that we need to do, I think, much more in South-South collaborations. And of course, I have, we here in Africa obviously have a lot of respect for our colleagues in India and what they have been able to achieve with, for example, a lot of the vaccines coming from India. And I think we need to do much more, and we are learning that we need to do much more to collaborate and to promote collaboration that would lead to faster results. Because I think too often the benefits of science, I think, in my opinion, they take too long to filter to the people that actually need them the most. And even for an epidemic like HIV, you still find that the communities that need certain interventions are the last ones to actually get those interventions. So we need to do much more to translate scientific innovation into actual impact on the health of populations. And so I think we need to do much more to promote scientific collaboration, particularly South-South collaborations.
Thank you. Thank you. Shahid, would you like to add India specific?
Yes, Uma. This pandemic has given us an opportunity to learn many things. Of course, many things about the virus, about the disease, but I feel more importantly, it has again emphasized that when knowledge is incomplete, it drives two kinds of responses. One is the response of fear, and we've seen a lot of that, not just in India, but around the world. And the other is to club together and start working together. Cooperation, and we've seen that as well. In my living memory in India, working over the last 30 plus years, I have not seen so many labs come together to do something and pull together to contribute. So in a way, fear is the bad part, but I think cooperation is the good side of it. Indians in general have a very fatalistic attitude towards life. And, you know, we come with this belief that, you know, the day we have to die, we will die no matter, you know, what. So, and some of that fatalism is also is also there to see on the streets today, even in cities like Delhi and Mumbai and Bangalore. You don't find people wearing masks. I know highly educated people who think that you know, this thing is all overblown and it's propaganda by Chinese companies, and you know, it's difficult to convince such people. And that brings me to the other point about communications, how good communication is important in all fields of life, especially at times when you are grappling with fear. And if there is one criticism I will make of world health bodies, it is that they did not very clearly communicate. They went into the details of science. Common man on the street is not interested in knowing what is the size of the droplet that carries how much virus. He wants to know whether mask will prevent or not. And the dilly-dallying that we've seen from WHO on masks has created a lot of confusion. The third factor is a factor of trust. Pandemics are not controlled as a law and order problem. Pandemics are controlled by building trust. Largely that approach has been missing from India and, you know, possibly other parts of the world as well. We converted a public health problem very early into a law and order problem, and that's not how you control pandemics. And my final submission is that pandemics are not just biological entities, they are also sociological entities. You cannot tell somebody the importance of prevention, importance of wearing masks or hand hygiene or physical distancing if the next person is worried about where his or her next meal is coming from. And as scientists, we have completely lost track of that. So that's partly because of our siloed education, as Thumbi was saying. So I think beyond biology, I think as scientists, our duty is also to look into the human aspects of disease. And these are some of the lessons that at least I have learned from this outbreak.
Thank you. So I'll just take one question from the audience, which is in the chat box. I'm sorry, we didn't have time for too many. And this is kind of a conglomerate from various questions from various people, but it's actually about the science of the concept of a global virome project. So viruses evolve and they change over time. They compete with each other. So there's a lot of ecology and evolution happening with viruses. So the question, I guess, is if we have this kind of a snapshot approach, while we can do predictions and we can try to make inferences, will it really help us in the long term to really catch something before it happens, given that there's so much of evolution in viruses and so much of change in general, like climate change or whatever? All of these systems are dynamic and changing. So maybe we can let Jonna take a stab at that.
Sure. It's a complex question, and I did try to answer one person's. I'm sorry I can't do all at the same time, answer all of your questions, but there are a couple points. First and foremost, I'd like to say, as we're doing the project, we're also, and this is what we saw with PREDICT, we're building the capacity in the labs to do the real-time detection. I almost think of those labs as they have the ability to do viral discovery and characterization as kind of building an immune system for the planet, because then even if you only get the snapshot in time of the first identifications in each ecoregion where you're working, you have labs that are equipped when there is a spillover event and you start to notice cases that can very quickly jump in and say, okay, what is this? Has it changed from what we looked at and what we identified before? The other piece is that many viruses don't change very rapidly. They change rapidly enough to stay pathogenic and to exist, to not get sort of beaten down by the host's immune systems, but they don't all, I think we have in our mind that they're, like these zombie viruses that are changing so rapidly that we won't know anything about the ones that are coming because we studied the ones previously. And that's just frankly not the case for most of these viruses. They do change, and we need to detect and monitor for those changes that might make them more pathogenic, but we can't even start to understand what those might be if we don't get out there and get the information. It's kind of a chicken egg scenario when people bring up this argument, well, it's not worth doing because you might miss one, or you might the virus might change. I'm thinking, oh my goodness, well, I would have liked to known about 1,000 or 2,000 coronaviruses from the region to understand more, and to be able to have a pipeline for vaccines already started, and to be able to have better diagnostics. So it doesn't make sense to me to say it's not worth having the scientific data, because this is the only way we start to understand which ones are going to be pathogenic and which ones aren't, and what changes to monitor for.
Thank you. So I think we should probably wrap up since it's 12:20. Sudhir, any final thoughts? Or?
Well, you cannot really have final thoughts, but it was an amazing 2 hours. Thank you all. I'm still processing them in my head and written a whole lot of notes. But maybe I could just point out 2 things in addition. You've talked about change, you've talked about collaboration, you've talked about interdisciplinarity, you've talked about trust. There are 2 points that you have sort of overlooked, and one is that we cannot ignore the nation state. The nation state is a reality. When crisis happens, you notice that borders come down at multiple levels. Borders come down at the level of the nation, borders come down at the level of communities. And this is something that as we're going ahead, we need to think about this particular aspect. The second is incentives. What are the incentives for prevention and public health? This is one of the biggest challenges in public health. How do you incentivize public health? And I'm hoping that we will have creative solutions to this when we come back. So this is, I'm not saying that what I said was more important or less important, but just 2 points that I thought I should add in addition to the absolutely marvelous 2 hours. So we will be back. We're going to do our homework, Uma, Shahid, all of us in India. We already have a lab in Africa, and so we will be talking to our colleagues there too. You will hear from us after we've done our homework. So thank you again for an amazing morning. It's probably night in California, but yeah, thank you for your time. Thank you.
Thank you so much. I'll be closing the call now, and please share the video which is uploaded onto YouTube, and make it as widely available as possible. Thank you and good day.